All right, my name is Phillip Kalaniopio, and today we will be discussing how we here at NAU are investigating the combinatorial effects of RAF inhibiting compounds in BRAF-mutant zebrafish. And I understand that's a very involved title, and every bit of that will be broken down throughout this talk. So to start, I want to give a brief overview of precision medicine as a whole. It is the most current and up-to-date way that scientists and clinicians try to attack very complicated diseases such as cancer. And so that begins with wet bench molecular biologists and cancer biologists investigating different oncogenes and tumor suppressors and different genetic causes of cancer or the progression of cancer. And so a lot of times they will do that using a model organism. We use the zebrafish, Danio rario. And after that, they validate whether or not different genes contribute to cancer, and if so, what drugs can stop those genetic mechanisms from contributing to cancer. After that, those drugs are then moved on to clinicians and clinician scientists, in our case, collaborators at the Translational Genomics Research Institute in Phoenix, who then start with small scale and move on to large scale human clinical trials. So the part that my group here at NAU plays is this left side, the classical cancer research side. So now that that's covered, I want to talk about melanoma for a brief second. It's a cancer of the melanocytes. So there's these cells that secrete melanin, which is a protein that causes certain people to be darker than other people. And that is the basis of race, essentially. So in 2019, 96,000 people were diagnosed with melanoma in the United States, and a little over 7,000 of those will die. So melanoma is a very aggressive cancer, and it is a very common cancer, especially in places with a very high UV index, such as Arizona, or anywhere in the Southwest, really. And so I'd like to call your attention to this figure from a New England Journal of Medicine publication, where it was a phase 3 clinical trial for vemurafenib, a novel kinase inhibitor and one that we use in our studies. And so this is a waterfall plot. And on the far right side, what you can appreciate is that the bars that reach all the way below the x-axis and reach minus 100 means that the entire tumor was eradicated with this drug. But the issue with this comes with the far left side of the graph, where some people received vemurafenib and their tumors grew. So we want to address that problem. But in order to address it, we have to understand what the underlying problem is. And that lies in the mitogen activated protein kinase pathway, or MAPK as it's referred to. And our protein of interest is right here, the RAF protein. There are 3 isoforms, ARAF, BRAF, and CRAF, where BRAF is the most commonly utilized isoform in melanocytes and in the epidermis in general. So there are 3 broad classes of drugs that we will use in our studies. That is the type 1.5 kinase inhibitors, which include vemurafenib, as I previously discussed, and dabrafenib. There are type 2 inhibitors, which include sorafenib, and then type 3 or MEK inhibitors, which are trametinib and cobimetinib. So the type 1.5 and the type 2 kinase inhibitors both work on different conformations of the RAF protein, whereas type 3 inhibitors work on the MEK protein, which is immediately downstream of RAF. Downstream of that is ERK, and ERK is the ultimate signal transducer in this pathway, meaning that ERK does a lot of jobs, and it phosphorylates and turns on many other transcription factors, some of which contribute to cell growth, cell proliferation, cell division. And these are what we know as the hallmarks of cancer: unrestricted and uncontrolled cell growth and proliferation. So when it comes to mutations in the context of melanoma, this was taken from another New England Journal of Medicine article where they categorized and sequenced these mutations. And what they found was 60% of mutations in human melanoma were in the BRAF protein. And looking forward to that, the BRAF V600E mutation, the one that we look at specifically, is present in 15% of all cancers, which is a lot in the context of cancer, and 60% of melanomas, as we described on the previous slide. But the interesting part is that 90% of all mutations in the BRAF gene are of the V600E nature. So what makes this mutation so advantageous for a cancer cell? Um, as you can see in the gene map and the protein map, so V600 or valine 600 refers to there being a valine, that amino acid, at the 600th amino acid position, which in this case lies in exon 15. Now, exon 15 is important because it encodes the activation segment, or the part of the kinase that is responsible for basically opening up and phosphorylating the downstream protein, which in this case is MEK. And so that activation segment lies within the kinase domain. And so this all contributes to how this BRAF mutation, this one single point mutation, a T is changed to an A, thymine to an adenine, which changes a valine to a glutamic acid. And calling back to biochemistry, valine is a hydrophobic amino acid. Glutamic acid is a hydrophilic amino acid. So V600 in its wild type or normal state tucks itself in a hydrophobic pocket. When mutated to glutamic acid, that hydrophobic pocket is opened up due to the hydrophilicity of glutamic acid. When this is open, that kinase is constituently active. That is to say, it automatically starts phosphorylating MEK if ATP is available, which in a normal mammalian cell, we know that it is. And that concept of the hydrophobic pocket being opened up by this point mutation can be illustrated with these red lines. On the left, you can see the inactive form of BRAF, and that is the wild type. There is a valine at the 600th amino acid position. But on the right, you can see that the conformation of this protein is drastically different, and it's in its active state. And this is characterized by not only the opening of the activation segment, but the change in amino acid to a glutamic acid at the 600th position, which you can see are, both of these amino acids are colored in red, the activation segment being colored in green. So this begs the question, how does this specifically, one little point mutation in the context of one seemingly insignificant protein, contribute to something as aggressive and dangerous as melanoma? And the answer lies in dimerization. And this is when one BRAF protein, or any protein really, combines with another protein. So two different monomers, or in this case, BRAF proteins, can combine and become one dimer. Now, this dimer transduces a signal downstream to MEK, as we saw in the previous figure. The classical gold standard drug treatment for melanoma right now is one drug to inhibit RAF and one drug to inhibit MEK. So the novelty of our studies comes from trying to use two drugs to inhibit this RAF or protein dimer. And the idea being that, as we saw in the New England Journal of Medicine waterfall plot, there is resistance to these chemotherapeutic drugs. And one of the ways that that happens is by an incomplete inhibition of the dimer. So that is to say, one half of the dimer is blocked, but the other half of the dimer can transduce a signal. So you still get some of this signal transduction downstream that results in cells dividing. And so you can see that right here in this figure. So our goal is to use two different drugs to inhibit this dimer, hopefully inhibiting both monomers that make up the dimer, resulting in no cell proliferation. And one thing I'd like to call your attention to is that these different parts of the kinase are flexible, which we're going to talk about in just a second. So the kinase is dynamic, and that is to say it can freely move from its inactive state to its active state. And there are two intermediates that it can go through, the CIDO or the CODI conformations. Those refer to the positions of the DFG motif, which stands for aspartate, phenylalanine, and glycine, or the alpha C helix. Those two are our big distinguishers for what position and what phosphorylative capabilities this protein has. So what I want everyone to take home from this slide is that this kinase is not stuck in one position or another. It can freely move from an inactive to an active state in a wild type scenario. So our hypothesis being, we hope to lock this kinase in one of the inactive states, preventing it from moving into the active state, and thus preventing signal transduction altogether. And so we accomplish this first by using rule-based modeling. in collaboration with researchers at Los Alamos National Laboratory. So on the left, you can see what goes into a model. I could spend an hour talking here about just the modeling side of things, but what it allows us to do is model how these drugs and how these proteins and how these mutants work in silico. And this is really advantageous for us because there is a lot of drugs and there is a lot of different proteins and a lot of different ways that these can interact. So we can simplify this by using rule-based modeling and using that to make predictions about what drugs should work and what drugs are worth our time in pursuing. On the right is an extended contact diagram, basically showing how all our rules interact with one another. And every square, every big, large gray square is a protein. So you can see RAS, you can see CRAF, BRAF, mutant BRAF on the far right, and then MEK and ERK. So this basically makes up the MAPK pathway, or the crux of it. And so you can see different domains and subdomains of these proteins, such as the MEK and RAS binding domains on RAF, or the RAF and ERK binding domains on MEK. And so every single part of these proteins are mapped and assigned a rule, and then different sites of phosphorylation or inhibition are mapped likewise. And so hopefully that will lead to furthering our understanding of how to basically silence this pathway and silence the BRAF V600E mutant. And so what you can see hopefully in this figure is that using one drug is not enough. You can hit 1/2 of the dimer, but that's not good enough to stop cell proliferation and division. So ultimately what you need to do is hit both monomers that make up the dimer and silence the dimer completely, which is the main hope of this entire project, is to inhibit the BRAF V600E homodimer with two different drugs that work in synergy with one another. So real quick, I wanted to talk about how these inhibitors work in the context of all the permutations that these isoforms can take. They can be in either both the CO-DO conformation, meaning the alpha C helix and the DFG motif are both out or in the inactive position. And then we also have the CI-DO or CODI, referring to one of either the DFG motif or the alpha C helix being in the active state and the other not being in the active state. And this is where we really want to trap this kinase. We want to prevent it from oscillating into the active form, which is the CI-DI conformation. And unfortunately, all type 1 inhibitors at this time are experimental. So blocking the active form of RAF isn't an option for us. What we have to do is block it really in that inactive state and prevent it from becoming active. And so I've talked a lot about synergy, and now I want to tell you how we're going to figure that out. We're going to figure synergy out through in silico methods or rule-based modeling, as I described a few slides ago. And then we will move to in vitro and in vivo methods in a stepwise fashion. So once something is validated in silico, we move in vitro, and once something's validated in cell culture, we move to the zebrafish. However, Ruklenko et al., 2018, the Cell Systems paper, found that a type 1.5 and a type 2 drug in combination does result in synergistic reduction in MEK signal transduction. And so what that means is you can see on the left side of this graph here, what lines constitute antagonism, or these drugs work against each other. Additivity, which has to do with basically 1 + 1 = 2. So drug 1 plus drug 2 equals a result, and we understand that, and that makes sense. And synergy, which is assessing synergism, has to do with the analogy I'd like to use is 1 + 1 = 4. We know that the sum is greater than the parts, and that is what makes this truly synergistic. They work together better than they work apart. And so just to drive this message home, 2 drugs is better than 1. And so one of the ways that we're going to do this in vivo is using Toll2-mediated transgenesis. So we will inject the Mini Cooper eGFP expression vector plus the Toll2 transposase into humanized BRAF V600E mutant zebrafish. So they are p53 null, which is a tumor suppressor, and they are homozygous inserts for the BRAF oncogene. And so what this means is that they're in a very compromised genetic scenario. So they are very prone to developing cancer. They also lack MITFA, which is melanocyte-inducing transcription factor A. So they're albino. And the injection of this plasmid back into the genome gives them the MITFA transgene back. But the MITFA promoter is driving GFP. So in our hands, we've seen that this plasmid can increase susceptibility to melanoma by about 40 to 50%. And it's mosaic. So you'll see that some parts of the fish will have large melanomas, whereas some will look totally normal, completely wild type. And you can see that in the middle figure here of a gross imaged fish that had to be euthanized due to the COVID-19 pandemic. We didn't have time to run a full drug trial, unfortunately, and I had hoped to have more data for this presentation by now, but unfortunately that wasn't the case. And on the far left, you can see that that is an image I took of the actual injections of a one-cell embryo. It's the same way that one would deliver the CRISPR-Cas9 system or other sorts of gene editing tools. And on the right, you can see an experimental schematic basically showing out our workflow. So the far left image can be corresponded to figure C on the right, and the middle image of the fluorescent zebrafish can be best identified with letter E on the right. And unfortunately, we never got to the oral gavage stage in this time frame, but once this pandemic kind of blows over and we're allowed to get back into the lab again, that is the top priority on my list. But it also kind of made this section of the presentation difficult because we were not able to do a full drug trial. So we were not able to give these fish this isoflurane and MS-222 mediated anesthesia, which is a new novel method for anesthetizing our fish because they need to be anesthetized for 14 days. And anyone who works with live animals knows that it's very hard to anesthetize them. And so we found a better way to do it. We just haven't been able to do it yet, unfortunately. But the top right figure will show you how much better this new combination seems to be than just using MS-222 on its own. And then on the bottom, you can see the oral gavage technique that we are also planning to use from the same authors that came up with that isoflurane MS-222 anesthesia method. And they use this to deliver known concentrations of their kinase inhibitor, which in their study they used vemurafenib, into the adult zebrafish. And on the bottom left, you can see an image of Dr. Salanga and I in the fish room working with our pneumatic microinjector and our microscope, things that we will use in these studies to gene edit or anesthetize or otherwise manipulate these test animals. I had also hoped to have some Western blot images to show today, but unfortunately, once again, due to the pandemic, I wasn't able to. But our hypothesis remains the same. By using synergistic combinations of drugs, we should inhibit BRAF so much so to the point that we see a decrease in phosphorylated and total ERK, which is downstream of BRAF. And you can see that in the leftmost image. This is a paper from the previously mentioned Ruklenko Cell Systems paper, where they used a gradient of doses of trametinib in combination with dabrafenib, so a type 3 in combination with a type 1.5 inhibitor. So it's slightly different than our hypothesis, but I think it gets the point across. That what they see is in a short exposure, high doses of dabrafenib and trametinib effectively silence the MAPK pathway. And when you start to use, so if you look at the very, very far left well, their control, they had no drugs. So I would assume that this was treated with DMSO or their vehicle control, has very large protein expression for phospho and total ERK, which makes sense. There has been nothing to inhibit these proteins, so they get expressed. I'd like to move to my acknowledgement slide, and I really want to thank the Salanga, Proper, and Posner Joint Lab Group. They've been awesome to work with. They've really helped me out a lot, and it's been a lot of fun working with them. I'd really like to thank the Flynn Foundation for funding this project. They really care about human health in Arizona, and specifically liked our project for how it tries to tackle melanoma in a different and novel way. Next is the Lewis Stokes Alliance for Minority Participation. They've funded me for 2 years. I've been a mentor. I've been a research scholar. They've sent me to a few conferences, and I can't speak highly enough about this program. Next is clinician scientists that we work with at TGen. They've been very instrumental in helping us formulate our hypotheses and really help get this project moving along. Next is Dr. Bill Lavicek and his group of theoretical biology and biophysics at Los Alamos National Laboratory. He is one of the experts when it comes to rule-based modeling, and especially with the MAP kinase pathway. So he's been instrumental in getting this project off the ground. And then finally, our collaborators at the Systems Biology Ireland and University College Dublin, who I mentioned a few times in this presentation, is Ruklenko and his group. And so with that, I will move to my works cited slide, and I thank you for your attention.